to introduce Ms. Cami Pearson. Cami received a Bachelor's of Science in Psychology from the University of South Dakota in 2018. She's currently a second-year master's candidate in Dr. Ehab Sarsour's lab, and her research investigates the relationship between aging and lipid metabolism. So the floor is yours, Cami.
Thank you very much for that introduction. As she mentioned, my name is Cami Pearson, and the title of my project is Decreases in G0, G1 switch 2 expression and Increases in lipid peroxidation, impair wound healing, and aging normal human fibroblasts. So we've had the opportunity during the last few days of the symposium here to learn about some interesting research. A lot of it's about specific diseases like cardiac disease, diabetes mellitus, and neurodegenerative disorders. And while these are all very different, one of the things that they all have in common, diseases like this, is that the risk for developing them increase with age. So that's one of the primary reasons why I'm interested in studying aging, because it acts as a predictive factor for many different health outcomes. The other reason I'm interested in studying aging is because it's one of the most inevitable things in life. My birthday was just the other day, so I got that reminder that none of us can avoid aging. And it's becoming more important for us to study aging because people are starting to live longer. In developed countries specifically, people are living longer as a result of improved public health standards. In the US, we see that there's a projected demographic shift that people ages 65 and older will make up an additional 10% of the population within the next 30 years. In addition to being associated with the development of certain diseases, we also know that aging alters the effectiveness for many health interventions that we have in place to treat these diseases. We often think about aging on the level of the organism, right? We know that at a certain point, aging becomes inversely related with quality of life. However, we can't forget that organisms are made up of tissues and cells, and these obviously also go through changes while we age. So aging cells lose their ability to repair damage and renew. So in our lab, we use a type of cell to study aging that's found in virtually every tissue and organ system in the body, and these are normal human fibroblasts. So these are responsible for things like the synthesis of extracellular matrix proteins, building and remodeling connective tissue, and regulating the volume and pressure of interstitial fluid. So here's just some examples of what these cells look like down here. And then previous work in our lab was done to understand what sorts of cellular processes underwent the most changes during aging. So RNA sequencing was used to evaluate global gene expression patterns. Genomics and metabolomics were then used to identify that lipid metabolism is the #1 pathway that's altered most during aging. So as you can see here, we have genes that are either upregulated or downregulated in the young, and those are almost completely reversed in the old. So lipid metabolism is a very complex process with a lot of different moving parts. One aspect that we're interested in studying in our lab is the GOS2 and ATGL pathway of lipid metabolism. So ATGL is adipose triglyceride lipase. This is responsible for initiating lipid lipolysis by catalyzing the release of fatty acids from lipid droplets by cleaving triacylglycerols into diacylglycerols and fatty acids. A co-regulator of ATGL is CGI58, and then GOS2, which is G0, G1 switch 2, acts to inhibit lipolysis by negatively regulating ATGL. So this brings us to our research question, which is, do age-associated changes in lipid metabolism contribute to impaired wound healing capabilities in normal human fibroblasts? So again, we're using these normal human fibroblast cells. We have all received from reportedly healthy individuals from a variety of different ages. So we have 20 cell lines ranging in age from 17 years up to 79 years. We also have samples that are taken from the same individual at different ages. And we've also included, male, female, white, and African-American individuals here. So the first step of answering this question is to determine what components of lipid metabolism change during aging. So to do this, we used Western blotting to measure protein levels, and this showed us that GOS2 decreases during aging. In comparison here, ATGL remains relatively consistent throughout the lifespan. So this shows us that there's some level of dysregulation happening during lipid metabolism during aging. So one component that we were interested in looking at is the downstream effects of excessive lipolysis, and this is lipid peroxidation. Lipid peroxidation is the oxidative degeneration of lipids. So the lipids that make up different cellular organelles are a really common target for reactive oxygen species. And so through a variety of, through a series of chemical processes, this lipid peroxidation can result in damage to the organelles that are made up of lipids. So at high levels, lipid peroxidation can lead to cell and necrotic death. So to test lipid peroxidation levels in these normal human fibroblasts, we use a stain called BOTA-PC11. BOTA-PC11 can measure the difference between oxidized toxic lipid peroxides and non-oxidized neutral lipids. So we use BOTA-PC11 in live cell imaging in order to visualize this. As you can see here, we have the oxidized toxic lipid peroxides in green and the non-oxidized neutral lipids in red. I think you can see visually pretty clearly that the accumulation of toxic oxidized lipid peroxides in our representative old sample is much greater than that of our representative young sample. But of course, to quantify this information, we also created a ratio that shows us that the oxidized lipid peroxides versus the non-oxidized neutral lipids. And you can see a pretty steady incline in that ratio relative to age. Now, one of the aspects of lipid peroxidation that we just talked about is that at high levels, it can be really damaging to cells. So lipid peroxidation can lead to the cells not being able to repair properly. So this is why we decided to also look at wound healing capabilities in normal human fibroblasts. We also know that wound healing in chronic wounds are a really classic hallmark of aging. So this is important for us to study. So to do this, we use a wound healing plate that shows us in real time as the cells migrate and proliferate to fill their wounds. So you can see starting out at time 0, we've taken these cells over the course of 6 days, measuring these every 8 hours. At time 0, they're all starting out initially the same place. Within 72 hours, you can see that our representative young and old sample have already initiated that wound healing process and have begun to migrate and proliferate to heal those wounds. You can also see that the older normal human fibroblasts down here, most of those cells are sticking around the perimeter of that wound. At the end of the time course, 144 hours in, 6 days, we can see that the young and the old normal human fibroblasts have completely healed that wound, whereas the older normal human fibroblasts, the density of cells that are within that wound space is still pretty sparse. So in order to quantify this information, we took normalized cell counts within the wound space from time 0. And what you can see here is that while initially they all start proliferating at about the same rate, you can see that about halfway through this divergence begins. And so you can see that up here at the top, we have cells that are below the age of 50, are younger cells. And these have a higher density of cell count, of cells within their wound space relative to the older cells, those that are above the age of 50 years. Another way that we wanted to look at this data is understanding the amount of time that was necessary for each cell line to migrate and proliferate enough to completely heal their wound. So taking this information, this number of hours, we were able to plot that out. And you can also see that that this is also increasing during aging relative to age. So in conclusion, just to reiterate some of the pathway information that we discussed, adipose triglyceride lipase catalyzes the release of fatty acids from lipid droplets. G0, G1 switch 2 acts to inhibit lipolysis by negatively regulating ATGL. We've seen that there's a decrease in GOS2 during aging, and so this leads to lipolysis being unregulated. And this excessive amount of lipolysis then contributes to an accumulation of toxic lipid peroxides in the impaired wound healing capabilities that we saw as well. So this is only a fraction of the sort of research that I've done. I have also done some causality studies using drugs that regulate lipolysis and other assays that help to validate the results that I've gathered thus far. But I'm only allowed 10 minutes, so if you're interested in hearing more about that, come to my thesis defense. In conclusion, many of the age-associated human pathologies that we see are driven by inflammation. So investigating these age-associated changes in lipid metabolism that contribute to pro-inflammatory phenotypes can help us to identify targets that can be exploited for preventative and interventional therapies. So I'd like to take a moment to acknowledge everyone who's assisted on this project because it's a lot of people. So especially Dr. Sarsour and his lab, our collaborators at the University of Iowa, the Zadie lab, my other committee members on my thesis committee here, Dr. Zadie and Dr. Singh, the Kansas City University College of Biosciences, and of course, Dr. White. Any questions?
So excellent presentation. Wonderful job.
Thank you.
Cami. My question for you has to do with the data on the increase in the peroxide accumulation as a function of age. So do you have any idea as to where this accumulation is occurring, whether it's in the cytoplasm or it's in the mitochondria? Any idea of where?
It's related to the mitochondria. That's where we often see the reactive oxygen species, but this toxic lipid peroxide can happen to any sort of cell membrane that's made up of lipids.
Right.
So,
because a lot of them happen in the plasma membrane,
Yep.
the lipid peroxides. So that was the first question. And maybe a good follow-up experiment would be to do like a co-localization. So do mitochondrial staining and see if the peroxides are
right.
present there. My second question has to do with, have you or anybody else looked at the antioxidant status of these old cells? Because it could, this accumulation of peroxide could be due to the increased synthesis of the peroxide or a compromisability of the cell to degrade them, by virtue of antioxidants, glutathione and such. So any idea if anybody has looked into that?
I think I have seen some research about it. I'm not positive off the top of my head. I do know that that's not something that I have time to do on my thesis. And I'm sure that if there are other students that come after me, they would be willing to work on that part of the project, because we're looking right now at just like one of the things that we intended to do originally was to see what feeding the cells free fatty acids would do. And so that's related to that also, but it's not something that's intended to do on my project yet.
Thank you.
Dr. Zaidi, I did that. I measured the antioxidants in aging fibroblasts.
There we go.